In the next two sessions, we will learn about the genetic code, how it was defined, and speculate on how it may have been sculpted by evolution. We will also cover types of mutations, including primary mutations in the coding sequences of genes, and both intragenic and extragenic suppressors of the primary mutations. We'll learn about polymorphic markers and how they can be used to track traits in organisms, including humans, when we don't know what the causal mutations are. Through all of this, I hope you will get a picture in your mind of genomic structure and how we can use that to our advantage as geneticists. Here, I'd like to review the concept of the triplet code that encodes the amino acids, following up on the work on R2 done by Francis Crick and discussed previously. In any coding sequence, there are three possible reading frames, but only one is the correct one. In either frames 2 or 3, although the letters are correct, the reading frame is not, so that the words make no sense. In the bottom example, we've introduced a nucleotide into the frame. As a consequence, the words read correctly up to the insertion, but after that, the reading frame shifts and the words can no longer be read. Similarly, when we remove a nucleotide, we would also go out of frame. In the R2 experiment, combinations of insertions and deletions were used to illustrate that the reading frame is 3 bases long. That is illustrated here. Relative to the wild-type sequence at the top, first 1, then 2, then 3 bases are inserted into the sequence. While initially out of frame, the reading frame is restored by insertion of the 3rd base. Depending on where the insertion takes place, the sequence will encode either additional or different amino acids around the point of insertion. Deletion of base pairs will have a similar effect. The R2 experiment told us that the code is 3 base pairs long, but did not identify how the 20 amino acids, which were known to compose proteins, were encoded. Neither did it explain how the theoretical maximum of 64 codons, that's 4 nucleotides arranged in all possible combinations, were used to encode the 20 amino acids. Nor was punctuation, the start and stop of translation, explained by these experiments. 2 key innovations led to identification of the code. The first was the ability to carry out translation in vitro using a cell extract containing ribosomes, amino acids, and tRNAs. The second was chemical synthesis of nucleic acids, RNA in this case, that could be used as artificial substrates. Thus, with input RNA, the cell extract would read the RNA and make protein. The first such studies used poly A, C, G, or U. This work led to identifying UUU, AAA, CCC, and GGG as encoding phenylalanine, lysine, proline, and glycine, as it was these amino acids that were incorporated into a peptide sequence. The experiment led to a Nobel Prize in 1968. H. Gobind Khorana was a chemist who also got the Nobel Prize in 1968, and he figured out how to synthesize dinucleotides and trinucleotides, allowing combinations of different nucleotides to be used to work out the code. In this example, alternating UC dinucleotides were shown to encode SER-LU proteins. Not only did this prove that UCU or CUC encoded serine or leucine, but it also proved that the code was not overlapping. Then someone figured out how to mix a known 3 base pair RNA with ribosomes and a tRNA. Doing this for all of the 3 base pair sequences using each C14 labeled amino acid allowed the code to be definitively worked out. Up to this point, all of this was worked out in vitro. Charlie Yanofsky showed that the in vitro predictions held in vivo, demonstrating that point mutations led to a predictable change in a single amino acid in the TRP operon in E. coli, which encodes the tryptophan biosynthetic enzymes. These studies then again showed that the genetic code is not overlapping, as only 1 rather than 3 amino acids were changed by a point mutation. Then, in studies in humans, it was shown that a single amino acid substitution could result in sickle cell anemia. This finding was important for two reasons. First, it showed that the code, which was identified in bacteria and viruses, appeared to be the same in humans, suggesting the near universality of the code, which we now know to be true. This means the code was evolutionarily early. Second, it showed that a genetic disease could result from a single amino acid substitution. Here is the genetic code, and a few things stand out. First is codon degeneracy, which means that multiple codons can code for the same amino acid. And most of these differ at the third position. Also, we can see that many similar amino acids differ from each other by a single nucleotide change. For example, the hydrophobic residues are in the first column. There is debate over whether this was the original design or whether it evolved over time through selection. This would protect proteins from point mutations by selecting for a code that would commonly result in substitutions by similar amino acids with a single base change. Finally, the code has punctuation, methionine as the start codon, and 3 stop codons. As an exercise, let's make a synthetic RNA by mixing U and G at a 3:1 ratio and then translate the resulting RNA population in vitro. Note that we have not specified the order of nucleotides, only their concentrations. We can then calculate the expected frequencies of each codon, that is, each 3 base pair sequence, accounting for all of the possible codons that can result from a combination of G and U. From the codon abundances, we can then predict the amino acid composition of the resulting proteins, yet another way to show which codons specify which amino acids, and illustrating the degeneracy of the code, with GUU and GUG both encoding valine, and GGU and GGG both encoding glycine. So the last piece of the translation puzzle was how the mRNA was translated into protein. tRNAs were predicted by Francis Crick, discoverer of the double helix structure of DNA, and he used structural considerations to predict tRNAs. The quote from him is, "I cannot conceive of any structure for either nucleic acid acting as a direct template for amino acids, or at least as a specific template. In its simplest form, there would be 20 kinds of adapter molecules, one for each amino acid, and 20 different enzymes to join the amino acid to their adapters." The predicted adapter molecules, the tRNAs, were isolated soon after, with subsequent identification of the aminoacyl tRNA synthetases needed to attach them to the amino acids. So we can stop the history lesson here and look at the anatomy of translation, all of which, as you know, occurs on the ribosome. The codons of the mRNA, in this case UAC encoding tyrosine, are recognized by complementarity between the codon and the anticodon of the specific tRNA that is charged in this case with tyrosine. Especially note the antiparallel orientation of the anticodon. We have discussed intragenic suppressors, that is, mutations that either reverse mutations to restore function or second mutations that compensate for the initial mutation. In the screens for suppressors, mutations in tRNA genes can also result. In this example, we have an idealized protein coding sequence and a mutation in the second codon, changing it from UAC tyrosine to UAG stop. Selection for suppressors resulted in a tRNA suppressor mutation designated as SU-, in which the UAG stop codon is now recognized by a mutant tRNA tyrosine with its anticodon changed from 5' GUA 3' to 5' CUA 3'. Now the tRNA tyrosine recognizes the stop codon and inserts tyrosine, restoring the function of the protein. You might think about whether the suppressor would have to put in the same amino acid or whether another amino acid would do. Also, could you use selection to suppress a pathogenic missense change? So here's a practice problem. The question asks whether by changing the anticodon of a tRNA could you change the sequence of the encoded protein? Here, the ACA anticodon, which would recognize a cysteine codon in the RNA, is changed to CCA. The tRNA is otherwise unchanged and would be charged with cysteine as the amino acid. However, the anticodon would now recognize the UGG tryptophan codon on the mRNA and insert cysteine instead.